All right, I'm going to go into presentation mode. Okay, hopefully everyone can see my screen and hear me. Thank you for the introduction. My name is Alex Rouhier, and today I will be defending my thesis entitled Evaluating Microshades Color Palette Accessibility for Color Vision Deficiency. My thesis advisor is Lisa Karstens, and my committee members are Michael Mooney, Annette Totten, Stephen Bedrick, and Tammy Martin. Today, I will explain the project, provide background information, highlight gaps in the research, review my specific aims, go over methods and analysis, discuss results and conclusions, and finish with the project's limitations and potential future direction. This project originated from two key questions. First, does the microshades color palette improve accessibility of the plots published in microbiome literature for those with color vision deficiency, which I'll refer to as CVD for the remainder of this presentation? And second, how can we make CVD accessible color palettes widely available to microbiome researchers creating plots for publication? Color vision deficiency affects approximately 8% of men and 0.5% of women. There are three types of CVD: protenope, deuteranope, and tritanope. Here on the right is a graphical representation of the three optical receptors we use to perceive color and how each is affected by different types of CVD. Protenopia is a green cone deficiency, deuteranopia a red cone deficiency, and tritanopia a blue cone deficiency. CVD can also be categorized by severity. There are three general severity levels: anomalous trichromacy, where one cone is mutated, dichromacy, where one cone is missing completely, and monochromacy, which is full color blindness. On the left, you can see this image of colorful crayons simulated to represent the protenope view, the deuteranope view, and the tritanope view. Both deuteranopes and protenopes have a hard time distinguishing between red and green, whereas tritanopes have more difficulty with yellows and blues. Next, let me introduce the Microshades R package, which is central to the project. Microshades was created by the Karstens Lab here at OHSU, and it provides two color palettes, one of which was specifically designed to be CVD accessible. Originally, this package was created to improve readability of common microbiome data visualizations in R, such as taxonomic relative abundance stacked bar plots. But the color palette can be applied to any type of plot. Here on the left is the Microshades CVD color palette, which is the focus of the project. The same palette is shown again on the right, but it's been simulated to show you how it would appear to someone with CVD. The top right palette is simulated in the deuteranope view, the middle in the protenope view, and the lower right in the tritanope view. As you can see, each of the 30 colors in the Microshades CVD palette are clearly distinguishable, even in these three CVD simulations. So why is CVD accessibility important? Scientific figures rely heavily on color to communicate information in a visual way. And when readers can't distinguish between colors in a figure, it affects their ability to understand and build upon the work of others. It's important to consider CVD accessibility in any form of written or visual communication, but particularly in scientific figures. Let me give a few examples. Let's say you are presenting work at a conference with an audience of 1,000 people. It's likely that around 40 people in your audience will have a color vision deficiency. Or let's say you are submitting a manuscript to a peer-reviewed journal for publication. If all three of your reviewers are male and of Northern European descent, there's a 1 in 5 chance that one of them will have a color vision deficiency. In this project, I focused on evaluating the accessibility of a common microbiome data visualization, the taxonomic relative abundance stacked bar plot. I chose this focus for three primary reasons: the growing popularity of the microbiome field, its links to several diseases such as IBS, diabetes, Parkinson's, Alzheimer's, and more, as well as the complexity of the microbiome data visualization. Microbiome data consists of highly dimensional genetic information from the bacteria, fungi, viruses, eukaryotes, and archaea that make up the human microbiota. Here's an example of a taxonomic relative abundance stacked bar plot, which I'll refer to as a taxobar plot for the remainder of the presentation. This plot provides a visual summary of all microbes identified in each sample and their quantities as a percentage of the whole. As you can see, there's a lot of information packed into this visualization. With over 200 taxonomic families represented here, it's almost impossible to tell which exact family a given color represents because the shades of each color are too similar. Taxobar plots play a critical role in the microbiome data analysis process. However, there are a few problems with current taxobar plots. Default color palettes use either a continuous spectrum of colors or a discrete set of colors, often with less than 10 color options. Both of these default color palette types are problematic for taxobar plots. Continuous color palettes make it difficult to differentiate between the individual colors from one taxa to the next when applied to a highly dimensional data set. In discrete color palettes, there just aren't enough colors to represent all colors in the taxobar plot. This means that colors are repeated, causing confusion when reading the plot and trying to identify specific taxa. And I'll show a few examples. Here are two taxobar plots created in Python with QIIME 2 showing 36 different taxa groups. The plot on the left was made using the default color palette, which is discrete and contains 8 different colors. All colors in the palette repeat at least 4 times, meaning that each one is representing 4 or more different taxa. The plot on the right was made using an alternate color palette option in QIIME 2, which is continuous. The individual colors in this plot are so similar, it's difficult to differentiate between adjacent colors. Here's another example of a taxobar plot created in R showing 25 different taxa groups. On the left, you can see a plot made with the default color palette, which is continuous. In contrast, the plot on the right uses the microshades CBD color palette to represent that same data. The circles in these plots are highlighting some of the lower abundance taxa that are really hard to see in the leftmost plot, but become clearly visible with the microshades color palette. This project had 2 main goals. First was to evaluate the accessibility of taxobar plots in the microshades color palette for those with CVD. And second was to prepare the microshades color palette as an official add-on to a popular microbiome analysis tool. To accomplish these goals, we needed to know: how can we assess CVD accessibility? How accessible are the figures currently published in scientific literature? And would any popular microbiome analysis tool allow us to incorporate the microshades color palette? So there are several relevant publications in the literature. Studies by Frayne et al. Published in 2015. Studies by Frayne et al. Published in 2015, Jambor et al. From 2021, and a recent publication by Angerbauer et al. Published last year. These studies use CVD simulations to evaluate accessibility, and they all established some level of inaccessibility in published scientific images and figures. These studies concluded with suggested guidelines to improve figure accessibility. However, journals seem to have either not adopted or not enforced CVD accessibility guidelines in their publications. Our study aimed to fill important gaps in current research around CVD accessibility, specifically for more complex data visualizations. The studies mentioned don't evaluate highly dimensional figures like taxonomic abundance stacked bar plots. Below is an example of the type of plot analyzed in prior studies. Now compare that simple plot with the taxobar plot here on the right, which communicates much more information and is inherently more difficult for someone with CVD to read. These studies also did not provide a CVD accessible color palette solution, which works for the high dimensionality of taxobar plots. Microbiome data requires a color palette with a greater number of discrete CVD accessible colors. Furthermore, journals are either not translating the guidelines suggested by prior studies into their publication requirements, or are not enforcing their requirements when reviewing manuscripts for publication. There are also gaps in functionality. QIIME 2's default color palette is problematic in terms of CVD accessibility, and the tool doesn't currently have a CVD accessible color palette as an alternative. QIIME 2's taxa bar plot function does not take custom color palettes as an argument. Colors can only be changed after the plot is generated using an interactive page which provides a limited drop-down list of QIIME 2 color options. This adds to the current difficulty of creating CVD accessible plots within the tool. For this project, we set the following specific aims. First, we aimed to evaluate the accessibility of the microshades CVD color palette in taxa bar plots for those with color vision deficiency. And second, we aimed to prepare the microshades CVD color palette as a QIIME 2 plugin. To accomplish the first specific aim, we surveyed scientists with experience in the biology or microbiome fields, asking them to rate the accessibility of taxa bar plots in both the original colors and also recolored with the microshade CVD palette. Our study consisted of 3 cohorts, a pilot group, a CVD cohort, and a full color vision cohort. The pilot group was made up of 5 full color vision volunteers who completed a pre-production version of our survey. And the purpose of this group was to verify the survey duration, the questions, and the overall user experience for the survey. The CVD cohort included 10 participants who were diagnosed with CVD, and these participants took the survey without any simulations. The full color vision cohort included 100 participants with full color vision, who were assigned to one of 5 subgroups. These subgroups were either given surveys without any CVD simulation, in the case of control group, or figures simulated to represent the type of CVD corresponding to that specific group. We collected responses from participants with CVD and full color vision simultaneously. Here's a visual representation of the built-in logic used to separate participants into subgroups. Participants were automatically assigned to either the CVD or the full color vision cohorts according to their response to the demographic question about CVD diagnoses at the beginning of the survey. Those with CVD took a version of the survey without any simulation, and those with full color vision were randomly assigned to one of 5 different groups: protanope, deuteranope, tritanope, monochrome, or control. The survey had 2 sections, demographic and evaluation. The demographic section consisted of questions asking about participants' sex, education level, and any CVD diagnoses or other vision impairments. The evaluation section had 3 types of questions for each scientific figure shown. Interpretive questions, which had participants interact with the taxa bar plot by asking things like, does this sample have a greater abundance of bacteria X or Y? Confidence questions, asking participants to rank their confidence level, and accessibility questions, asking participants to rate the figure as either accessible, borderline, or not accessible. We provided definitions of these ratings to ensure consistency between participants. Here's an evaluation question from our survey, showing a taxa bar plot with questions below asking about plot interpretation, confidence levels, and plot accessibility. The interpretation question here required respondents to really slow down and interact with the figure before rating its accessibility. The survey used figures selected from published microbiome literature. In January, we performed a literature search in the ScienceDirect database using terms like microbiome, human, taxonomy, and stacked bar plot. From these results, we reviewed publications in search of taxonomic relative abundance stacked bar plots that met our figure requirements and selected the first 10 accessible figures. In order to ensure consistency, all taxa bar plots were cropped down to a single bar in the plot and resized to a standard height and width. These modified plots were recreated with the microshade CVD color palette using an automated plot recoloring function written in R. The survey was created in REDCap the OHSU approved secure web application for creating and managing online surveys and databases in compliance with HIPAA and IRB requirements. The survey was reviewed and approved by both OHSU's IRB panel and the REDCap team before launch. We provided survey links to participants, which they used to navigate to our survey. And all data was collected, stored, and managed within REDCap to ensure data security. We did not collect any identifying information from participants within the survey, such as names or email addresses. Participants were recruited via conference announcements, posted flyers, tweets, and direct email, and all data analysis was completed in R. To accomplish the second specific aim, I wrote a Python package that runs functions from the microshades R package in a background instance of R and registered the function with the Chime2 suite in accordance with the Chime2 plugin guidelines. This function includes, this wrapper includes microshades functions such as the color mapping function and the plotting function. To write the Python wrapper, I used commands from the rpy2 package to run microshades functions in R. This method prevents inconsistencies between functionality in R and Python with microshades because both packages are based on the code written in R. However, it does mean that users will need both Python and R installed in their workspace to use our Chime2 plugin. We chose to integrate the microshades color palette with Chime2 for several reasons. Chime2 is one of the top 3 most popular analysis tools for microbiome data, with modular packages for customizable workflows. This tool is free and open source, with an extensive, involved community of users. The Chime2 suite has comprehensive documentation and tutorials. And importantly, for the purposes of our project, Chime2 allows outside developers to create plugin packages. Overall, Chime2 checked off our 3 main requirements. It's widely used in microbiome research, it's friendly to novice programmers, and it allows for plugin packages. Next, I will present the results of our microshades CBD accessibility survey. First, let's take a look at some of the participant demographics. Over 200 people agreed to participate in our survey, but not everyone completed the survey. Our final dataset included 100 participants with full color vision and 10 participants with some type of CBD who all completed the entire survey. The full color vision participants had been previously assigned to 5 different groups: control, protanope, deuteranope, tritanope, and monochrome. This table here shows the distribution of participant sex within each group. Notably, the tritanope group only had 15% male participants, a much larger skew than the other groups. The participants in the CBD cohort were all male, but this was not surprising as CBD affects males far more frequently than females. So now let's take a closer look at the CBD cohort. Among the 10 CBD participants, 60% reported a diagnosis of deuteranomaly, a form of anomalous trichromacy that affects the red cone. As this is the most commonly diagnosed color deficiency, it makes sense that most of our participants with CBD reported having this type. Protanomaly is another form of anomalous trichromacy, but this type of CBD affects the green cone. Tritanomaly, one of the rarer forms of CBD, affects the blue cone. One participant reported other and wrote in red/green color blindness, which is a generic term encompassing both deuteranope and protanope CBD categories. Next, let's go over the results from the control group and the simulated groups from the full color vision cohort. We start by looking at the accessibility rating results, specifically the average percent of plots rated not accessible in each group. These average percents were calculated by finding the percent of plots rated not accessible for each participant and then averaging this across all participants in each survey subgroup. In Figure 1, we see that across almost all simulated survey subgroup participants, on average, rated the microshades plots as not accessible less frequently than the same plots in their original colors. We would interpret this reduction as an improvement in plot accessibility with the microshade CVD color palette. Now let's dive a bit deeper and break these results down by survey question. In Figure 2, we see that the accessibility rating results varied quite a bit from one question to the next. Excuse me. This figure represents the percent of participants who rated the plot in each question as not accessible. Now we would expect to see that the plots in the original color palette, represented here in blue, would more frequently be rated as not accessible when compared to plots with the microshade CVD color palette. And several of the survey questions match our expectations, including questions 1, 3, 6, and 8. However, questions 2, 4, and 7 did not match our expectations. Also, please note that any missing bars in these plots simply represent a value of 0%. So to test our hypothesis, we used McNemar's exact chi-squared test to analyze the accessibility rating results for each survey subgroup. This test takes binary paired data and compares the number of discordant pairs between each of the two categories. In the case of our data, each original taxa bar plot is paired with the same plot in the microshade CVD color palette, and the binary outcomes are either accessible/borderline or not accessible. For example, in this contingency table, we see that there are 19 occurrences where paired plots were rated as accessible or borderline in the microshades color palette, but rated not accessible in the original color palette. By focusing primarily on the not accessible ratings and combining the accessible and borderline ratings together, this data becomes binary. This table summarizes the effects of the microshade CVD color palette on taxa bar plots in our survey. In the left column, we see that the percent of microshades plots rated as not accessible, as well as the percent of plots in their original colors rated as not accessible. Again, the majority of groups saw a reduction in the percent of not accessible plots when reading those in the microshades color palette. The second row of this table shows results for the all simulated group, which is made up of the agglomerated results from the deuteranope, protanope, tritanope, and monochrome groups. We found that the microshade CVD color palette significantly decreased the number of plots rated as not accessible for both the agglomerated all simulated group and the monochrome group, which represents the most extreme case of CVD. Next, we will look at the interpretation accuracy results, specifically the average percent of plots that were incorrectly interpreted in each group. In Figure 3, we see that across all survey subgroup, participants on average interpreted the microshades plots incorrectly less frequently than the same plots in their original colors. We would interpret this reduction as an improvement in plot readability with the microshade CVD color palette. In Figure 4, we see that the interpretation accuracy also varied from one question to the next. Again, we would expect to see that plots in the original color palette, shown here in blue, would more frequently be interpreted incorrectly when compared to plots with the microshade CVD color palette. And again, most of the survey questions matched our expectations, including questions 1, 2, 3, 6, and 8. But questions 4 and 7 did not match our expectations. And similar to the accessibility ratings we discussed before, we also used McNemar's exact chi-squared test to analyze the interpretation accuracy results for each survey subgroup. We found that the microshades CVD color palette significantly decreased the number of plots interpreted incorrectly for almost all survey subgroups. Finally, let's look at the results for the CVD cohort. At first glance, the CVD participants' plot accessibility ratings, shown here in Figure 5, don't seem to align with the results seen in our simulation groups. In Figure 5, we see this unexpected trend where plots with the microshade CVD color palette have been rated not accessible more frequently than plots in their original color palettes overall. And again, we can look at the results broken down by question. Figure 6 shows the percent difference in not accessible plot ratings between the microshades color palette and the original color palette. These results are shown for the CVD cohort here in teal and the agglomerated simulation group here in pink. As in previous results, a negative percent difference represents a decrease in the number of plots rated not accessible, and therefore an improvement in plot accessibility with the microshades CVD color palette. Just as with the simulated results, the CVD cohort results varied quite a bit from one question to the next. Interestingly, the CVD cohort and the simulated groups are in agreement in terms of directionality of change for each question. They only differ in magnitude. In Figure 7, we see that for some of the CVD participants, the microshades plots were less frequently interpreted incorrectly. However, it seems that the color palette had no overall effect for several other CVD participants. For most of the taxa bar plots, the microshades CVD color palette decreased the number of incorrect interpretations. And like with the accessibility rating results, we see similar trends within each question for the CVD cohort and the simulation groups. We did not perform statistical tests with the CVD cohort due to the small number of participants. So to better understand the variability between questions, let's take a look at one of the survey questions where we saw those unexpected results. Survey question 2 asked participants to determine if bacteria I is present in the sample. The correct answer to this question is no, bacteria I is not present. With both the simulation and the CVD participants, we observed that the microshades color palette worsened the plot's accessibility ratings, but improved its interpretation accuracy. These observations seem incongruent until we review the taxa bar plots and the questions posed. Here we see that the plot's original color palette on the left used two very similar colors of blue to represent different taxa. However, the blue represented in this sample is actually bacteria E, not bacteria I. The answer is much clearer in the microshades version of the plot, and most participants rated the original plot as accessible, believing the answer was straightforward, but then interpreted the plot incorrectly. When comparing our results to those of prior studies, the percent of plots rated not accessible by our survey participants was far higher. For example, a recent study by Angerbauer et al. Found that 16% of scientific plots included in their survey were not accessible, while we found that more than 30% of taxa bar plots were not accessible. There are several reasons why our rates of inaccessibility were so much higher. First, our study used dichromatic CVD simulations, which are the most extreme versions of each CVD type. Second, our study had participants review plots with much higher complexity, with up to 18 different colors in each plot. Third, our plots did not include any titles, axis labels, or text descriptions. And prior studies have shown that text descriptions and labels can be extremely important in plot accessibility. Finally, we designed the questions in our survey to specifically target colors in the original taxa bar plots, which were most affected by the CVD simulations. This study did have a few limitations. First, CVD simulators don't perfectly reflect the CVD experience. They're a close replication of what someone with CVD would see, but deficiencies in color vision do fall on a spectrum and they vary from person to person. Our results show that trends among the simulated groups at the survey question level do align with the CVD group, but the differences in magnitude of change between the original color palette and the microshades resulted in incongruent outcomes overall. Second, some survey groups were underpowered due to the lower than expected effect size. These groups in particular would have benefited from either a longer evaluation section of the survey with more taxa bar plots or additional participants. Third, some of the taxa bar plots included in the study contained visual artifacts due to the process to compress JPEG images for publication. These artifacts appeared as bands of color, which may have affected both plot accessibility and readability. Fourth, this study only investigated the microshades CVD color palette, not the microshades package itself. The taxa bar plots in this study were not created using the microshades package and therefore did not exactly reflect the package's color and legend organization. And finally, the Chime 2 plugin has a limitation in that users will need both Python and R installed in their computer to use it. For future research, I would suggest increasing the number of participants in each survey subgroup, using PNG images as opposed to JPEG, and limiting the variability between survey questions by generating the taxa bar plots from raw data with the same default color palette for original images, and then the microshades package for recolored images. In summary, we detected significant improvement in plot accessibility with microshades color palette for simulated groups as a whole, and in particular the monochrome group, which represents the most extreme CVD simulation. We also detected significant improvement in interpretation accuracy with microshades color palette for the all simulated group, as well as 3 of the 4 individual groups, deuteranope, tritanope, and monochrome. Our control group did not show a significant improvement in figure accessibility or interpretation accuracy with the microshades color palette. This result was expected as participants in this group have full color vision and they looked at plots without any CVD simulations, thereby reducing the beneficial effects of the microshades CVD color palette. With the CVD cohort, we did not see overall trends indicating improved accessibility with the microshades CVD color palette. However, we did observe some improvement in plot readability for the CVD cohort as a whole. And when looking at the breakdown by question, the microshades color palette had a similar impact on both CVD and simulation groups. In other words, questions where the microshades color palette improved accessibility or readability for the simulated groups, the CVD cohort also saw an improvement. However, the effects for the simulated groups and the CVD cohort differed in magnitude. These results indicate that the microshades CVD color palette may have a measurable positive effect on plot readability if we were to have a larger group of CVD participants. Now, I won't discuss my second aim in too much detail as it's very technical, but I'll give a brief overview of the accomplished tasks for this aim. So we have defined the system requirements, written a Python function which wraps microshades functions from R and runs a basic microshades pipeline. We've written the Chime 2 plugin setup file and created necessary documentation files for the package, and also tested all Python wrapped microshades functions and submitted a pull request to the microshades Chime 2 plugin. Added a pull request for the microshades Chime 2 plugin to the Karsten's Lab GitHub. Now let me review some of the key points discussed today. Not all scientific figures in published literature are accessible to those with CVD, but the microshades package has been shown to improve plot accessibility and readability in published taxa bar plots with CVD simulations. Some types of CVD may benefit more from the microshades package than others, and future research should include a greater portion of CVD participants to better understand this. Chime 2 is an ideal platform for our new microshades package to maximize usage and awareness in the microbiome research community. I wanted to give a big thank you to my thesis advisor, Lisa Karstens, for helping me develop and complete this project and providing guidance along the way. Thank you also to my thesis advisory committee members, Michael Mooney, Stephen Bedrick, and Annette Totten, for all their input and feedback. and also to Tammy Martin for acting as my external committee member from outside DMICE. Finally, thank you to the Carstens Lab for developing the microshades package, and Erin Dahl in particular for her help with some of the visualizations in this presentation. All right, thank you everyone, and are there any questions? And here are my references. All right, I'm going to stop sharing my screen.